And we'll move now to the last talk, Genes, Cells, Cardiology, Dr. Moshe Flugelman from the Carmel Medical Center in Haifa, Israel.
Thank you, Mr. Chairman. I would like to thank the organizers for giving me the opportunity to present in this prestigious session. The topic of my presentation is genes, cells, and cardiology, and more appropriately is be aware of the omics, genomics, proteomics, metabolomics, and so on. This is my disclosure. And in my presentation, I will start with a brief introduction, how our genes expressed as proteins. Then I will show you our work on genes and cells used as a therapeutic agent for induction of arteriogenesis. And I will finalize how cardiology can or will be changed by progress in molecular and cellular biology. This is a drawing of Gregor Mendel, who is the founder of genetics. And Mendel showed us that there are hereditary determinants, traits, of a particulate nature, which we refer now to as genes. He also showed us that the law of segregation, which refers to recessive and dominant traits, and the law of independent assortment, which relates to the fact that our genes are inherited independently. In 1953, Crick and Watson showed us that the DNA, which encodes the genetic code, is a double helix structure shown here with a sugar phosphate backbone. Oops. And pairs of bases, adenine and thymine, and cytosine and guanine, the sequence of which is really the code for the proteins that the gene will express. Based on the knowledge that was acquired after several centuries, the central dogma of molecular biology was that our code, the genetic code, is in the DNA, which is transcribed to a molecule of an RNA, and then the RNA is translated in the ribosomes into proteins. We know now that only 1.5 to 2% of our genome is considered to be protein coding part, which is genes. And we also know that we have about 20,000 genes, similar to chimpanzees and mouse. And of interest is the rice have 37,000 genes. But over the last 25 years, with increasing knowledge expansion, it's a matter of explosion of knowledge, we know that there is an increasing complexity of the central dogma of molecular biology. It's no more DNA transcribed to RNA and then protein. We know that there is a transcriptional regulation of the DNA with the three-dimensional DNA structure, epigenetic changes, protein-DNA interaction, and RNA-DNA interaction. Also, in the RNA processing part, there is a lot of regulation. We know that a single gene can code several proteins based on the exons that are expressed. And we also know that RNA can serve as a regulatory element and also as an enzyme. And finally, if we go to the post-translational modification, we know that once a protein is formed, it can produce more than one protein phenotype by different folding and chemical properties. All this knowledge was also used for gene therapy. And now we know that gene therapy can be used in monogenic diseases to replace defected gene or to express a therapeutic gene. And the process is outlined in this cartoon. We take a normal gene, we put it in a vector, and then the vector knows how to get the gene into the nucleus where the gene is transcribed and later on translated. The most known success of gene therapy is in babies that were born with no immune system, usually due to a single gene mutation or single defected gene. And these children must be grown in isolated, sterile environment, and this is why they are known as the bubble boys. The process of treating these children and babies, and many of them been treated in this way, is to harvest the bone marrow cells, then introduce a normal gene to replace the defected gene and reinstate the bone marrow cells in these babies. And several dozens of babies now in Europe and USA have recovered from the disease and conduct normal way of living. Our research focus for the last 25 years was on angiogenesis. Angiogenesis occurs mostly in the embryo, and it starts with binding of VEGF, this protein, to receptors on endothelial cells and formation of a primitive tube. This primitive tube can be reabsorbed, and it's leaky of proteins and cells. This oversimplified cartoon comes to an end when angiopoietin 1 binds to Type 2 receptor, and the second layer of cells stabilize or mature the blood vessels. In case of capillaries, it's a pericytes, and in case of arteries and veins, it's smooth muscle cells. We also now know that genes can be grouped or classified to families according to their function. With the discovery of VEGF by Napoleon Ferrara, a lot of excitement in the community was initiated, and many researchers started both animal and human experiments with delivering of VEGF protein and VEGF gene to ischemic tissue, mostly in the heart and the legs. And they've all been able to show that the number of capillaries was increased significantly. Unfortunately, even when we increase the number of capillaries by tenfold, blood flow can be increased only by 30%, and therefore no alleviation of this ischemia can be achieved. If we want to improve or alleviate ischemia syndromes, we have to increase the number of arteries, and then we can increase the amount of blood flow even by tenfolds. We treat today patients with arterial occlusion either with PTCA or stents, time-honored procedure. I'm a cardiologist, bypass surgery, but I would like to draw your attention mainly to this angiogram of a patient with an occluded superficial femoral artery. You can see that the patient developed from the side branch a very significant network of collateral arteries. In many patients, this collateral network will be sufficient and the patient will be asymptomatic. But in many other patients, patients have an insufficient blood flow through this collateral network, and they will suffer either from claudication, breast pain, unhealing ulcer, or frank gangrene. We focus with our work on enhancing this collateral circulation by injecting autologous venous, endothelial, and smooth muscle cells that express the key genes in angiogenesis, Ang-1 and VEGF, to induce arteriogenesis. In order to accomplish the therapeutic goal, we have to show first that we can deliver angiopoietin 1 and VEGF to primary human endothelial cells and smooth muscle cells, and shown here, this is the typical cobblestone morphology of endothelial cells, and we've been able to deliver angiopoietin gene to these cells, as in the smooth muscle cells with the typical hills and valley morphology that we've been able to deliver the VEGF gene. In order to test the functionality of these activated cells, we conducted this in vitro angiogenesis three-dimensional spheroid model. This is as close as you can get in the lab on the bench to creating new blood vessels. Here we tag endothelial cells with a yellow fluorescent color and smooth muscle cells with a green fluorescent color, and then we see them on methylcellulose non-adherent membrane. The cells form this spheroid, and when we embed this spheroid in collagen, after 24 to 72 hours, the cells start to sprout and produce structures that are very similar to blood vessels. In this series of experiments, we've been able to show that when smooth muscle cells express VEGF and endothelial cells express angiopoietin 1, there is a robust sprouting of two cell types, green cells and yellow cells. And this is as close as you can get in vitro to production of double layer or double cell blood vessels. We then tested the effect of gene transfer on the gene expression of the human cells, and we've been able to show that when we introduce a single gene into these cells, we activate many families of genes families of cell motion, blood vessel development, blood vessel morphogenesis, vascular development, cell adhesion, and cell proliferation. All these families can contribute to remodeling of existing arteries or create new arteries. We then took this concept and tested it in a hind limb rabbit model. We tested it in a very large number of rabbits. We isolated endothelial cells and smooth muscle cells from the external jugular vein of the rabbit and then injected it after gene transfer to the hind limb, and we tested both blood flow and muscle perfusion, and we've been able to show that only with the two cells that with endothelial cells and smooth muscle cells that express angiopoietin-1 and VEGF, we've been able to increase flow to nearly the normal blood flow levels and improve perfusion in the ischemic limb. We also showed with CT angiography that a very robust collateral circulation developed in the treated animals, unlike the control animals, and the developed collaterals were stable for more than 6 months. We then carried on and continued to human experiments. Of course, there is a huge amount of safety data, and the concept is outlined in this cartoon. We isolate under local anesthesia a small segment vein from the arm of the patient. Then we isolate smooth muscle cells and endothelial cells. We transfer the gene, we expand the number of the cells, and we transfer the cells to the cath lab, where using a simple diagnostic catheter, we inject the cells proximal to the occlusion of the main artery. The cells are carried by blood flow to the collateral circulation, and we show that they stick to the vessel wall and exert their repertoire of genes and proteins, and the collateral circulation is expanded, and a larger amount of blood can be delivered to the distal organ, distally to the occlusion. We conducted the first study in 12 patients in the USA. It was safety, but we also tested for efficacy in University of Michigan and University of Pennsylvania. And as shown here, this patient with limiting claudication that failed conventional therapies had a longer time before claudication started and also could walk longer time on a treadmill. This is one example of an angiography in a patient that had a failed bypass graft with EPTFE graft and occluded the stent. We see a very nice demonstration of the below-knee arteries, and which are supplied by a very well-developed network of collaterals of the therapy that were secondary that resulted from injecting the genetically engineered cells. A second study that we conducted in Israel, again safety and efficacy, but this time in sicker patients with critical limb ischemia, and shown here is the follow-up of 18 patients with Rutherford 4 and 5, which refers to rest pain and unhealing ulcers, and we see a very high survival rate, amputation-free survival rate at 2 years of more than 60%, and in comparison to published data, this is a very high survival rate. This is an example of a patient that have amputation of his toes, unhealing surgical wound, and 6 months after injecting cell injection have a completely healed ulcer. Now, in the last part of my presentation, I will talk about the future. Futurism is an art movement that started in Italy in the 1920s, and just using this single painting, we can see that predicting the future is truly impossible. This is the artist's view of an airport, and we all know that the airplanes look different and the airport look different. So I will stick to facts. I will show you 2 recent hypotheses that relates to how omics will influence our practice. So when we talk about omics and cardiology, we have to talk about monogenic disease versus polygenic diseases. We have to talk about mutation in coding and non-coding regions, and we have to talk about gene-environment interaction, or the science of epigenetics, which means after-genetics or over-genetics. So going back to the complexity of the central dogma, we have to look at how transcription is regulated, RNA is processed, and proteins are regulated. So after completion of the human genome in 2003, we had the blueprint of life, and for us cardiologists is the anatomy of the DNA. But what was missing is the physiology. So the function of vast majority of the 3 billion bases is still unknown, and with a joint collaborative effort of many centers and intellects, the Encyclopedia of DNA Elements ENCODE project mapped systematically regions of transcription, transcription factor association, chromatin structure, and histone modification. This data enabled to assign biochemical function to 80% of the genome, in particular outside the well-studied protein coding region, the genes. The publication of the results of the ENCODE project was accepted with enthusiasm, but also with very significant criticism to several scientific points, but nevertheless, we should look at what they published. This is the architecture of human regulatory network derived from the ENCODE data. We see the relationship between 119 transcription factors and microRNAs, and I brought this graph to show you that the complexity of the regulation of transcription, and also to show you another way, the co-association between different transcription factors in this gigantic matrix. Red denotes a very high association, and white denotes very low association. And just looking at this matrix, we can realize how complex regulation of expression and transcription is. Finally, I would like to take old data and to show you how we can use new tools to understand this data. This is the classical publication by Tavia Gordon from Public Health Reports in 1957. He showed that when we look at cardiovascular mortality of Japanese men, it was very low, but when they immigrated to Hawaii and continental USA, it was doubled, but was still less than half of the white people the same year in the USA. And the assumption was that the genetic pool of the Japanese men had less predisposition for atherosclerosis, and that the environment doubled their predisposition to atherosclerosis by moving to a different country. The study of epigenetics may provide explanation how environment interact with our genes. There is an exponential increase in number of publications related to epigenetics in general and also in cardiovascular medicine. And if we look at the structure, three-dimensional structure of DNA and protein, we know that both regulate gene expression. This is our chromosome and then chromatin, and we see that the DNA is tangled around histones. Histones are proteins, and we also know that these small tails, the chemical composition of which really determine whether the gene will be expressed or not expressed. We also know that methylation of regulatory elements shuts off a gene. We see here the CG islands in the non-coding gene, and we know that transcription factors can bind to these sites and then they open gene expression, while methylation of CG islands can close off the expression of several genes. And I would like to show you two hypotheses how environment affects these genes. This is a recent publication by Ling and Ron from Sweden that claim that regular exercise changed the genome while the DNA methylation pattern in human skeletal muscle and adipose tissue. So regular exercise altered DNA methylation and microRNA expression, and therefore improve metabolic phenotype and decrease the risk of disease. A second hypothesis, far more complex one, comes from Spain by Corella and Ordovas, and they claim that Mediterranean diet through transcription factor 7 like 2, TCF7L2. It's a very important polymorphism that predicts type 2 diabetes in Europe. Modification of expression of this transcription factor due to a high Mediterranean diet can downregulate or upregulate several genes and therefore affect our risk for stroke and cardiovascular morbidity and mortality. So when we talk about the future, it's a very good place to remember Sir William Osler, the father of modern medicine, at bedsides. This is a series of very impressive pictures showing him observing, palpating, and auscultating, and then thinking about the patient. And the question comes whether our future cardiologists will need clinical skills. And my personal view is that they will still need clinical skills to take care of patients, but they also will have to be aware and educate and study about the different omics, such as epigenome, genome, transcriptome, proteome, and metabolome, and the interaction between the different omics that will give us eventually with a very complex and computer heavy analysis, the cardiovascular phenotype. So in summary, we know that functional understanding of gene regulation and transcription will provide deeper understanding of pathology and potential therapies for mono and multi-gene related diseases. I've shown you that convergence of cell and gene therapies may provide new venues for regenerative cardiology, and epigenetics may provide the explanation how environment affects gene expression and regulation. And finally, we all have to be aware of the omics as it will take a significant part in our practice. I thank you for your attention with the beautiful picture of Haifa.
Any questions for Professor Flugelman? If not, thank you, Moshe. We had a really very exciting session with 4 excellent lectures, sometimes provocative, but all of very high quality. I would like to thank my co-chairs, Professor Pinto, Professor Sahar, and all the audience, and thank you very much.